Hi again, welcome to our continuing series of videos. This one's on reproduction, but for part two, looking at DNA. We said in the last video that the point, if you really think about it, for reproduction is to pass on this molecule DNA from parents to offspring, or in the case of asexual, from parent to offspring. So what is DNA and why is it so important that there's a whole mechanism called reproduction to pass it on? If you think about this, these couple of questions, if you consider what type of information is stored in your DNA and how different is your DNA from anyone else's, it may give you a little bit of a clue. And just to give you this idea, the DNA of two humans, the same gender, is 99.5% identical. So if you're a male, your DNA is 99.5% the same as other males, and females obviously the same thing. So only 0.5% of your DNA, a really small part, actually contributes to or is responsible for your unique traits. So what type of what type of information is actually stored in the DNA? We have a story to go back to then to kind of get to that question. DNA, of course, is a molecule, deoxyribonucleic acid, and we're not going to dwell on its structure too much. We'll look a little bit more in class at this, but it's made of sugars and phosphates and four bases, and you know them as adenine, guanine, cytosine, and thymine. The DNA was discovered back in the 1860s, actually, in the 19th century, but we didn't know what it did, didn't know why it was important, and it seemed pretty simple. With just four bases, it didn't seem like it'd be a very complex molecule, and it took until 1953 before we really appreciated what DNA did. You see here, this is James Watson, and this is Francis Crick, and they're showing you a model of DNA. They constructed this model based on a number of scientific experiments, and this is now the model for DNA, not the model. That is, this is now, we know DNA structure. So it took until 1953 before we really appreciated it. So if you do the math, it's only been in the last 60 years or so that we've really begun to understand what DNA does and why it's so important. But we're going to back up for a second. Let's go back to chromosomes. We'll be looking at chromosomes in lab, and we'll talk more about chromosomes and what they do. But a chromosome count for humans is 46. That is, in a normal human with the normal allocation of chromosomes, there's going to be 46. A chromosome is simply a long piece of DNA wrapped around proteins. So in cells that have DNA, as not all cells do, but in cells that have DNA, if you look in the nucleus, and I lost my pen, if you look in the nucleus here, in this nucleus there's going to be 46 pieces of DNA that are wrapped around proteins, and this constitutes our chromosomes. If somebody has a condition like Down syndrome, then they're going to have 47, and they're going to have 47, they'll have one extra of a particular type. We won't get into the details of this too much, but we actually can number all these chromosomes. We number them based on size, and there's two chromosome number ones. There's two chromosome number twos. There's two chromosome number threes, etc. So we go all the way to two chromosome 22s, and that gives us 44 chromosomes. Then if you are a female, you have an X and an X. If you're a male, you have 44 chromosomes and an X and a Y, and that Y is what makes a male a male. So I'm coming back to this point that when we're talking about chromosomes, we're talking about long pieces of DNA wrapped around proteins, and there's 46 identifiable ones of these in each cell. And why do you have really two sets of 23? Because the sperm had 23 chromosomes in it, as did the egg. Put them together to form a zygote, and we're back to 46. So once again, let's make sure we understand how DNA and chromosomes all relate. If we look inside the nucleus and we find these 46 chromosomes, if we begin to unravel these, we're going to get all this structure and all these little balls here, These red balls here are the proteins I mentioned before. That's what DNA wraps around. Ultimately, if we pull the DNA loose from that and we look very carefully, we'll see A's, T's, C's, and G's. These are the bases, adenine, guanine, cytosine, and thymine, that join DNA together. So ultimately, DNA wound around the histones, wound up some more, tightly associated in a chromosome. That is what the relationship is between DNA and a chromosome. So then what's a gene? You've heard of this term, genes. Aren't they the same thing as chromosomes? Not really. A chromosome, as we've just seen, is a huge piece of DNA. A particular sequence of that, a particular sequence is a gene. So what do we mean by a particular sequence? If we were to unravel DNA enough to where we can see all of this, again, now we're down to the A's and the T's and the C's and the G's. A particular segment, and they're showing you, it's kind of hard to see here, the yellow, but this segment right here, they're calling this a gene. This gene is a set of instructions for making a protein. And we're going to talk more about proteins in the next video. But a protein is going to be something like melanin or something like insulin or something like keratin. It's a protein you make that's important for your structure or for your functioning, like enzymes. So a gene is a set of directions for making a protein. There could be a gene that goes from here to there. There could be a gene that goes from here all the way over to there. We have 30,000 of these genes on our chromosomes. That is, we have 30,000 sets of directions, if you will, to make a human. And let me get it out of the way. If we look at these bases, adenine, thymine, guanine, cytosine, it's actually the sequence of these bases, the exact order of these bases, that spells out the directions to make a protein. And we'll deal with that a little bit more in the next video. But if we consider what genes really do, they control the production of proteins, and I gave you examples before of insulin and melanin. Here we're seeing a not so great example of what can happen. If we have someone who's a smoker, like benzo-A pyrene is a chemical found in coal tar, in tar that we associate with cigarette smoking. This chemical is a mutagen. It actually damages the DNA. In this case, it damages the DNA that controls a gene that controls how fast that cell divides. So I'll try to say a little bit better. The gene itself is a gas pedal or brake. In this case, this is gene P53, which happens to be a brake pedal, and we don't really worry about that details, but it controls how fast cells divide. If you have a chemical that basically comes in and damages the brake, then this cell is not going to know when to stop dividing, and it will produce a tumor. So that's how we can associate benzo-A pyrene with cancer. So damages to the genes that control cell division, ultimately that's what cancer is. Either they damage the genes and make them speed up too fast, or they damage the genes that keep them from slowing down. Either case, you're going to have a disaster. If your car has a gas pedal stick, you're going to drive too fast. If the brake pedal breaks, you're going to drive ultimately too fast. So in the case of certain chemicals, and we happen to know this one, it's going to damage the DNA in a very specific spot, which damages the ability of that cell to control its cell division. So as we're just about wrapped up here, I just want to mention the bases one more time and how DNA makes copies of itself. We mentioned the A's and T's and C's and G's, the adenine, thymine, cytosine, and guanine. And the reason this is so specific here is that adenine really has two parking places where it can bond to something else, and it happens to bond to thymine because thymine also has two parking places where it can make bonds. Cytosine and guanine happen to have three. So that's why A's and T's always pair up in the DNA, and C's and G's always pair up in the DNA, because that's exactly how they can pair up. They can't really pair up any other way. Why is this significant? Oh, I forgot I had that. Why is it significant is that DNA can copy itself because it has this very direct pairing relationship. It says here, if you know half the DNA molecule, you know the other half. If you know one half is A and T, the other side must be T and A to match up. And as you'll see here in a second, let me go ahead and get out of the way here. As you'll see in a second, this is how we can pass on information from one cell to the next, and really from one organism to the next. It's because this exact copying mechanism of DNA. So if you look here, we have our original molecule, A bonds to T, C bonds to G, T to A to T, G to C. During DNA copying phase, and we'll look at that a little bit later, in one part of the cell cycle, the DNA is going to copy itself to get ready to put one copy into one new cell and one copy into the other. The light blue here is showing you the new version of DNA is being made, but notice A bonds to T, and A bonds to T, C to G, C to G. Since G and C can only bond to each other whenever G is exposed, like you see there, the only thing that can bond to it is a C rather. And you see the same thing happening here. Ultimately, I get two molecules that are exactly alike, AT, AT, CG, CG, etc. So when I look at this view of it, if I look at DNA can unwind itself and ultimately can produce two identical copies, that is really, really, really huge. So that every cell that divides is going to produce two cells that are each going to have a set of directions for running all the cell processes in those new cells. That's if everything goes well. So just to recap, video 1 and 2 here, video 1 is ultimately we have to think about reproduction as a process for copying and dividing up new DNA, or for making sure DNA gets sent to the next generation through an offspring. Here, you see a little bit of what DNA is so important. DNA controls or produces the traits that we need to survive. Video 3 will give you just a little bit more detail about that, and then we'll wrap up with an assignment. Thank you very much.